Welcome back all. In this section we are discussing about nitrogen fixation. Uh, the nitrogen fixation is the enzymatic conversion of gaseous nitrogen into ammonia. There are two types of nitrogen fixation: biological nitrogen fixation and non-biological nitrogen fixation. Non-biological nitrogen fixation is the physiochemical conversion of atmospheric nitrogen to nitrogen oxides or ammonia. It is affected by ionizing phenomena such as cosmic radiation, lightning, thunderstorms, etc. The high electric energy released during these breaks the triple bond of nitrogen and liberates free nitrogen atoms. It also brings about the photochemical fixation of free nitrogen atoms with the oxygen or hydrogen of atmospheric water to form nitrous and nitric oxides and ammonia. And nitrous and nitric oxides get hydrated with the atmospheric water, forming nitrous and nitric acids. And rainwater will bring these acids and ammonia to the soil surface. And soil nitrates and NH3 will be utilized by plants. So, non-biological nitrogen fixation. The biological nitrogen fixation is it is carried out by some living organisms called nitrogen fixers. Nitrogen fixers include some cyanobacteria, fungi, and lichens. They are falling under two groups: free living nitrogen fixers and symbiotic nitrogen fixers. Free living or non symbiotic. Example for non symbiotic nitrogen fixers are bacteria like acetobacter, bacillus, and rhodospirillum, etc. Aerobic bacteria like acetobacter and anaerobic like clostridium, photosynthetic like chlorobium, rhodopseudomonas, and chemosynthetic like thiobacillus, desulfovipro, and fungi like yeast pilullaria, and blue-green algae like nostoc, Annabina oscillatoria, etc. And Annabina is the genus of nitrogen fixing blue-green algae with bead-like or barrel-like cells and enlarged spores found as plankton in shallow water and on moist soil. There are both solitary and colonial forms. Colonial form resembles the nostoc family. In the temperature latitudes during the summer, Annabina may form water blooms. The symbiotic nitrogen fixation. The symbiotic nitrogen fixers include some bacteria, cyanobacteria, actinomycetes, etc. And the bacteria include some species of rhizobium like azorhizobium. They are found in the legumes of root legumes, and several species of Klebsiella living in the leaf nodules of some tropical plants such as Psychosia and Cyanobacteria. An example for these symbiotic species of Annabina living in the root nodules of Azolla. Azolla is a water fern, and some species of nostoc living along with certain fungi, and the actinomycetes like Frankia living in the root nodules of non-legumes like Casuarina, Myrica, etc. And the fungi and algae. Other example of symbiotic nitrogen fixes. Symbiosis is coined by D. Barry, and symbiotic nitrogen fixation account for more than half of the nitrogen used in agriculture. The members of genus Rhizobium and related alpha proteobacteria in association with the leguminous host plant will fix nitrogen. And the root associations, legume nodules. The association of nodules with roots of leguminous plant is one of the oldest reports in microbial ecology. In the late 1600s, Marsilio Malpighi made extensive drawings of legumes showing roots with well-developed nodules. Bacteria are specific for a legume species, and formation of the root nodule is the result of unique differentiation process. And the steps involved in the formation of root nodules. The legume root secretes specific chemicals known as flavonoids. Example for this is tryptophan, and bacteria will absorb and metabolize it into produce IAA, and these signaling molecules attract the rhizobia growing in the rhizosphere. This is the first picture in this. This is the root nodule. The flavonoids also induce transcription of node genes in the rhizobial genome to produce lipocytin oligosaccharides called node factors. The node factors is also known as root hair curling factors or nodulation factors. The plant root recognizes the chemical structure of the node factors, and these lipocytin oligosaccharide molecules are taken up by legume receptor kinases. The node factors activate the plant hair roots, and this recognition is responsible in part for the specificity between the host and legumes. The symbiotic bacteria attach onto the root hairs and enter into the root by a process called root infection or infection thread formation. And the fifth step is the bacteria produces lipopolysaccharide capsule that enables the rhizobia to evade plant defense systems and enter the root by a structure designated as the infection thread. Phytohormones are produced by bacteria to stimulate root cell division, and the plant root expands quickly to produce a nodule. The bacteria in the root nodule adjust to the low nitrogen environment and the reduced oxygen level in the nodule. In some cases, the bacteria differentiate to produce rounded cells or cells with Y shape. These differentiated bacteria are bacterioids, and these cellular units are important in fixing nitrogen. It's a structures of formation of root nodule. In this picture, the root hair curling factor is found, and the infection thread, this white portion is the infection thread, and is the pink colored are bacterium, and the infection thread is moving to the host cell and releases the rhizobia, and rhizobia were developed there, and Y-shaped cells. And another example of symbiotic nitrogen fixation in actinomycetes include Frankia. It's known as actinomyces, and these found in Frangia and 8 non-leguminous host plant families. This bacterial association is called actinomyces, and Frangia fixes nitrogen particularly in trees and shrubs. In stem nodulating rhizobia, they are found in tropical legumes. These nodules form at the base of adventitious roots branching out of the stem just above the soil surface. They contain oxygen producing photosynthetic tissues. Examples are Isorhizobium colinodens form nodules on legume Cispania rostrata. It's a picture. And the third one is fungal and bacterial endophyte. Specialized fungi and bacteria can live within some plants as endophytes. Clavicipitaceous fungi form systemic fungal infection in which the endophyte grows between the plant cortex cells. Plants infected with these endophyte may be less susceptible to attack by chewing insect due to presence of alkaloid produced by them. So fungi invaded into the cell, and Rhizobium leguminosum uh, trifoliforms a nitrogen fixing symbiosis with clover, can also form a endophytic association with the rice roots. Not all endophytic relationships are mutualitic, some are parasitic. Parasitic castration is an example for this. And other is Agrobacterium. Agrobacterium tumefacius is an alpha proteobacterium, causes Krogol disease. It results in the formation of tumor-like growth in plants. The genes for plant infection and virulence are encoded on Agrobacterium tumefacius plasmid called Ti plasmid. Some bacteria causes some diseases also in plants. These are the example for them. Wildfire disease of tobacco, uh, and halo blight of bean, citrus blast, leaf spot of bean, blight disease of rice, and there are so many different examples and symptoms and their pathogens. Pseudomonas syringae produces wildfire disease in tobacco, and the symptoms are spots and blights in the leaf. So this is the end of our section. Thank you.